Welcome back friends. Today in this video tutorial I will discuss with you about ELISA. It is called enzyme linked immunosorbant assay. It is a type of immunological technique that is widely used to identify different type of antigen and antibodies. It is very specific type of technique. Here we can isolate a particular antigen and antibody, and also we can study its quantitative level. Here, as the name implies, enzyme linked. You know very well what are enzyme. Enzyme are the biomolecules that enhance the rate of reaction. And here we used a substrate. The substrate impart color in this reaction. When it react with enzyme, it produce product that is colored, and the occurrence of the color estimate the presence or absence of various type of antigen or antibodies. The color imparting color producing substrate are also called chromogenic substrate, and we used various type of enzymes in ELISA. Commonly used enzyme are beta galactosidase, horse radish peroxidase, and third is alkaline phosphatase. Next is the type of ELISA. There are three types of ELISA. First is indirect ELISA, second is sandwich ELISA, third is competitive ELISA, and they have different type of protocols in this three type of ELISA techniques. The first type is indirect ELISA. Here is a diagrammatical protocol of the ELISA, that is indirect type of ELISA. Here what is done, we use a well or cavity slide in which we have artificially designed antigen. They are appropriate to the antibody that is to be detected, and the well are coated with the antigen. Or here we can identify antibody, specific antibody, because for the antigen the antibody will be specific. Here what we do, we use antigen coated well, then we wash it with distilled water, then we add specific antibody to this type of antigen, and what will happen there, on addition of antibody the cross reaction will occur and antigen and antibody complex will form. Next we wash the unbound antibody with distilled water and the immobilized complex is produced. Then we add secondary antibody. They are enzyme linked. In the Fc region of secondary antibody, the enzyme is linked, and the secondary antibody fuse with the primary antigen and antibody complex. Then we again wash, and we wash out unbound secondary antibody. Then we add a substrate that upon reaction with enzyme produce a color, and the type of the color produced identify the antibody we have isolated. If there is no color produced, there will be no antibody in the serum, and we can detect presence or absence of the infection with this type of color producing mechanism. This is very helpful in detection of HIV antibody. In the HIV infection, we can easily done this test to identify the antibody produced during the infection. If there is no color produced, the HIV test will negative, and if there is color is occurring, the test will be positive. Second type of ELISA is sandwich ELISA. Here we identify the particular antigen in this technique. In the first well, there are well is coated with the antibody, and here we are adding serum that is containing the antigen. Upon addition, cross-linking occur and antigen and antibody complex is formed. Then we wash the unbound antigen or wash out. Then we add secondary antibody that is enzyme linked, and here is sandwich like structure is produced. Hence its name is sandwich ELISA. Here the antigen is sandwiched between primary and secondary antibody. Then we again wash with distilled water and the unbound antibodies are washed out. Then we add a substrate that upon reaction with the enzyme will produce a color, and the production of color will estimate we have the presence or absence of the antigen. Hence, what we do in the sandwich ELISA, we identify antigen, particular type of antigen entering in our body, we can identify with the ELISA technique, that is sandwich type of ELISA. The third type is the competitive ELISA. As the name implies, there is a competition between the antibody and antigen for their binding. Firstly, we use incubated antigen and antibody outside. Outside the reaction, we incubate antigen and antibody and we produce antigen and antibody complex. Then we add antigen antibody complex with the antigen coated well. Here the another type of antigen is coated, and here is the higher concentration of this antigen as compared to the bound antigen. Then what will happen? There will be competition among the occupancy of epitope between paratop. Then we wash out the unbound antigen and antibody will be wash out. Then we will be adding enzyme linked antibody and the substrate will impart the color, and the color impart will show the binding of antigen is successful. If there is no binding of the antigen with the antibody, the color will not be produced. So what is the purpose of the ELISA? ELISA is very cheap and economical technique of detection of antigen and antibody. We can study the level of infection from the initial stages. We can identify the particular antigen or antibody, and also we can estimate from this type of immunological technique whether there is infection in our body or no infection. Here is the principle of the color production. The color is the estimation level of antigen and antibody. I hope friend this video tutorial is helpful for you to understanding the ELISA technique and its various types. If you like this video, like my channel and share it. Subscribe my channel. If you have any query about it, you can comment in comment box. Thank you very much. Have a wonderful day.